in this seminar. Thank you very much, Darrell. I appreciate your your time to give a lecture today. The Dr. Darrell Colbert received PhD from the University of Guelph in Canada, working with Dr. Anthony Clark on bacterial cellulose digestion. Dr. Colbert completed a first postdoc with Bertie Svensson at the Technical University of Denmark, working on surface binding sites in carbohydrate active enzymes. Second postdoc with Nicole Koropatkin at the University of Michigan, working on the structure and function of bacterial and starch-degrading enzymes. After started as assistant professor at Penn State University in 2017 in the Department of Food Science, working on the bacterial digestion of resistant starch in the human gut. Thank you very much, Darrell, for your time, for accept this invitation. I appreciate it. Thank you.
Yeah, and thank you for inviting me. I'm really happy to get this opportunity to share some of my work in my lab with you on resistant starch degrading bacteria in the human gut. So in my lab, we are generally interested in this interaction between diet and the gut microbiota. And my favorite aspect of this to work on is resistant starch. And so there are, of course, trillions of bacteria that are inhabiting our intestinal tract. And so all these bacteria need something to eat. And so there is mucus that we are producing in our intestinal tract that's lining our intestinal tract, providing something of a barrier between our cells and these bacteria, while also serving as a bit of a food source for them. So these are glycoproteins that many bacteria in the human intestinal tract are able to use for growth. But of course, they would like to eat other things as well. And of course, they get access to the food that we consume on a regular basis. And so while there's this sort of base level metabolism of the mucus layer, the majority of bacterial energy extraction and growth happens on the food that we eat. And so when we start out in life, that food is often milk from our mothers, or perhaps some sort of formula that's made to simulate that. And that leads to selecting for bacteria that are good at utilizing things like the lactose and human milk oligosaccharides that are present in there. But then we reach a point where we start weaning off of that diet and start bringing in more complex food sources. And so then as our diet diversity increases, so too does the diversity of our gut microbiota. And then eventually we reach a point where we kind of get to a more stable microbiota, where we have a certain range of species of bacteria that have colonized our intestinal tract. And what will happen is, as our diet changes from day to day or week to week, the relative amounts of those bacterial populations also fluctuate depending on what nutrient sources are available to them. But it's pretty hard to change actually which bacteria are there. So when we try and have things like probiotics that we input into the system, they can have an effect transiently while we are taking them, but it's very difficult to get them to establish in the gut and become permanent members of that gut microbiota, because we have relatively stable compositions of that microbiota over long time frames. And so to a certain extent, we kind of have to work with the microbiota that we get. So we can manipulate it to have a different metabolism perhaps, or to have different relative abundances of the species that are there, but it's very difficult to add new bacteria to it. So when we're looking at how that diet is shaping the gut microbiota, fiber is perhaps the most important part of that diet for shaping the bacterial population. And so bacteria have the capabilities of breaking down and utilizing a wide range of carbohydrates. Humans are much more limited. So, we can break down lactose, or we can break down sucrose. We can use most types of starches, but other things like cellulose, chitin, resistant starches, arabinoxylans, carrageenan, all these other potential carbohydrates that are in our diet, we don't have the enzymes to break them down. But there's a wide variety of bacteria in there, and some of them can break down these different sources of fiber and use them as an energy source. And this turns out to be one of the key things that shapes that composition of the gut microbiota. Then these bacteria in our intestinal tract can have a number of health influences. And the way that they're able to exert influence throughout the body, despite being localized to the gut, is that the metabolites that they produce can have a number of effects throughout the body. So that can be pieces of the bacteria, like the LPS or cell wall layer that are released, or the bacterial DNA, or it can be fermentation products from when they are growing on fibers or amino acids, or they might modify tryptophan in the gut, or they might modify bile acids. And then these modified bacterial metabolites can then interact with a number of different receptors in our intestinal tract and have effects throughout the body. If we focus in on particularly the effect of these short-chain fatty acids, acetate, butyrate, and propionate, which are the main bacterial fermentation products when bacteria in the gut are growing on carbohydrate sources like dietary fiber. And these can have a number of effects. So they can interact with various receptors, they can lower the pH and have antimicrobial effects against pathogens. They can influence the immune system through histone deacetylase inhibition, or through signal transduction through these various receptors. And butyrate in particular is the preferred energy source of these colon cells. So as it gets absorbed up here, they can have these effects with these receptors. Most of the butyrate gets used up by these intestinal cells. More can then go and be transported to the liver, where it can be further metabolized. And then by the time you get to the blood that's circulating around, very little of that butyrate is left. And so it has its primary effect here in the intestinal tract, but because of this signal transduction and immune effects, it can have influences throughout the body. So now let's talk a little bit about starch and what happens to it when it goes into the gut. So starch, of course, is just made up of the monosaccharide glucose, and it is joined together into two different types of molecules, amylose and amylopectin. So the amylose is just a linear chain of the alpha-1 4-link glucose, whereas the amylopectin has that linear backbone, but then also alpha-1 6 branch points coming off of there. Oops. Now, those linear portions of the amylopectin or in the amylose can come together. They naturally kind of have a helical form to them with this alpha linkages that are present. And as they come together, they form these double helices. In turn, those double helices can join together into crystalline arrays. And so when plants are making starch, you get alternating layers of crystallinity, where you've got these packing of these double helices, and regions of amorphous character, where you have these branch points in the amylopectin. The crystallinity that can form is generally in two different types, the A type and the B type. The A type is what we see in things like corn and wheat normally. The B type is what we see in things like tubers like potatoes. You can also have a C type, which is a mixture between these A types and B types. So human enzymes, if we cook up the starch to gelatinize it, so break down all that crystalline structure, they degrade that very well. If it is still in its crystalline form, if it's A type, the human enzymes still have an ability to degrade it, though more slowly. So this acts more as a more slowly degraded starch. But there can be other factors, such as having that B-type crystallinity, which make our enzymes not able to degrade that starch. And so it becomes what we call resistant starch. So there's different ways that you can have starch be resistant to digestion. What they all have in common is it's starch that makes it through the small intestine to the colon, to the site of the vast majority of the bacteria in our intestinal tract, where they can be degraded and used as energy sources. So we have that B-type crystallinity, which gives us our type 2 resistant starch. But we can also have a type 1 resistant starch, where, say, the starch is in a whole grain form, and so it's physically inaccessible to the enzyme. So there's, like, say, a bran layer or cell wall material that's blocking the enzymes from getting at those starch granules. We can also have retrograded starch, type 3 resistant starch. So in this case, the starch has been cooked and gelatinized, but over time it's been allowed to cool, and then crystallinity can start to reform. And so the amylose will tend to crystallize relatively quickly, the amylopectin more slowly over time, but as this recrystallizes, it again tends to form this B-type crystallinity, and the human enzymes again are not good at digesting it. We can also make chemically modified starches, and these are type 4 resistant starches. So typically through, say, cross-linking reactions. So the initial starch could have been an A type or a B type. But then once you cross-link it, you now make it more resistant to digestion. So you can take, for instance, a type 2 resistant starch and chemically modify it to make it even more resistant to digestion. So for instance, if you take just regular potato starch and consume it, you get about 60% of that raw potato starch makes it through to the colon. So we say that that has 60% resistant starch. If you took that same potato starch and chemically modified it, you would up that to higher levels. So you can make it more resistant, so more of it survives through to the colon. You can also have type 5 resistant starch, which is where we have things complexed into the amylose chains, which change the structure of it to again make it less digestible by the enzymes. So all these things kind of have different physical properties to them, but they all have in common that they are able to survive through transit through the small intestine to make it to the colon. And so what we kind of want as someone who is consuming that resistant starch to get benefits from our gut microbiota at the end is to have those short chain fatty acids being produced by members of the gut microbiota as they're fermenting that resistant starch, and particularly butyrate formation, which is thought to be the most beneficial of these short chain fatty acids. So if we look through the gut microbiota, you can find starch-degrading bacteria relatively commonly through the various taxonomic groups that are present. So there are starch-degrading Bacteroides, there's starch-degrading actinobacteria, there's starch-degrading proteobacteria, there's starch-degrading lactobacilli and bacilli and clostridia. But much like the human enzymes, most of these enzymes from these bacteria have trouble with the resistant starches. And so similarly to the humans, they can degrade the non-resistant starches, but are stymied when it comes to the resistant starches. There are a few organisms, though, Bifidobacterium adolescentis within the actinobacteria and Ruminococcus bromii within the Ruminococciaceae, that are able to efficiently degrade and utilize even these resistant starches in the gut. And so there is that capability existing within the gut microbiota, even though it's relatively rare. And it's possible that there's other species that we have not yet identified. I'll speak a little bit more about that later on. Now, we want to get that butyrate production. But so now we have to look at where butyrate production is coming from in the gut microbiota. And there's some other bacteria that can, say, produce butyrate via amino acid fermentation, but mostly we're concerned with butyrate that's coming from carbohydrate fermentation, as that seems to be by far the main source of butyrate in the gut. And so that butyrate being produced through carbohydrate fermentation is pretty much coming through the Clostridia group. Members within the Lactospiraceae, Rumicocciaceae, and Clostridiaceae. Now, not all members of those groups produce butyrate. There's sort of select members within each of them. And if you look within the Rumicocciaceae, so you'd see that Ruminococcus bromii falls in here. But unfortunately for us, if our goal is to get butyrate from resistant starch, Ruminococcus bromii does not produce butyrate. Others in the Rumicocciaceae, like Bacterium Prasnitzii, do, but bromii does not. And then our other one, Bifidobacterium adolescentis, within the actinobacteria, is primarily a lactate-producing bacteria. So we have butyrate producers in the gut, we have resistant starch degraders in the gut, but unfortunately they don't seem to overlap. And so if we are going to get butyrate from resistant starch, then we need some sort of cross-feeding interaction to be taking place between members of the gut microbiota. So we have a Ruminococcus bromii and Bifidobacterium adolescentis that are able to degrade resistant starch. Bromii produces mainly acetate as a fermentation product. Adolescentis produces mainly lactate. They can each produce some other things as well. Then we can look at our sort of groups of butyrate-producing bacteria. So there's the Eubacterium rectali and Roseberia group of bacteria. Many of these members are starch-degrading bacteria, but again, do not degrade the resistant starch. And they have some ability to consume acetate. So that's one product coming from the bromii. And potentially they could, any longer oligosaccharides or bigger chunks, but more soluble chunks of starch that are coming off during digestion by bromii and adolescentis, they could also further break down and produce butyrate during fermentation. Other bacteria like Bacterium Prasnitzii don't have any starch-degrading enzymes on their surface, but they can grow on maltose and glucose that could be released from the resistant starch by these bacteria, as well as some of the acetate to produce butyrate. And then there's this group, Anaerobutyricum halei and Anaerostypes hadris, that are able to, again, grow on maltose and glucose, but not longer pieces of starch, but can also utilize lactate and acetate to produce butyrate as well. So there's a number of potential interactions that could be taking place to allow that digestion of resistant starch to result in butyrate production. So if we look a little bit at that Eubacterium rectali and what it's doing with starch. So again, it's not going to break down that resistant starch, but it can grow on things like amylose and amylopectin and non-resistant starch granules. And it has a couple of enzymes on its surface, particularly this one large enzyme that has a number of different carbohydrate binding modules. So these are separate domains that are part of this protein, and they are dedicated toward binding to carbohydrates, and particularly these are starch-specific ones. So utilizing them, it's able to break down these starch polysaccharides, releasing mostly maltotetrose and maltopentose, so 4 glucose and 5 glucose unit long oligosaccharides. The other enzyme specializes in breaking down those short oligosaccharides that come from that enzyme into maltose. And then there's transporters for taking both the longer oligosaccharides and the maltose, and another one for taking in glucose. So it's able to grow on a pretty wide range of starch digestion products, but not the resistant starch directly. Other well-known starch-degrading bacteria in the gut include Bacteroides, species such as Bacteroides beta iota omicron. And they have a little bit different of a system. So they have this amylase on its surface, and then separate binding domain containing proteins that associate with it on the surface for, again, binding to these more soluble pieces of starch, degrading them, taking them in through an outer membrane transporter, then having periplasmic enzymes for further breaking it down, importing into the cell, and fermenting it. Now, in this case, the Bacteroides are generally not producing butyrate. They would produce things like propionate or succinate, for instance. So in some respects, they are competing with the butyrate producers for these starch digestion products. If we now kind of move on to these resistant starch-degrading bacterium, Bifidobacterium adolescentis, we start to see a couple of changes. One is that there's more of these starch-degrading enzymes. So it has 4 main ones that it produces on its surface, one of which is this big one that has 2 different catalytic domains and interspaced by some more of these carbohydrate binding modules. So in the case of both Eubacterium rectali and the Bacteroides, they just had this amylase component on their surface. But now we see for the first time one of these pullinase enzymes in one of these bacteria. So these pullinases are responsible for the debranching of the starch. So they break down those alpha-1,6 linkages. And so this activity is lacking in a lot of the other gut bacteria. So that's one difference about Bifidobacterium adolescentis from the others. Another is the diversity of the carbohydrate binding modules that it has. And in particular, it has this relatively unstudied to this point family called CBM74. So this is, you can see I depicted it a little larger than the other ones because it's bigger than your typical carbohydrate binding module. But in the preliminary studies that have been published so far, it seems to be better at binding to resistant starches than do the typical starch binding CBMs. And so that might be another key component that Bifidobacterium adolescentis has as well. Now, if we look at the other resistant starch-degrading bacteria, Ruminococcus bromii, we see an even more complex system. So here we have 8 different enzymes involved. And what you can see is there's a lot more domains going on here and some more accessory proteins as well. So we still see our carbohydrate binding modules, including the CBM74. We see both amylases and, in this case, 2 pullinases in the system. And then the extra layer of complexity here is in orange, we have these docrin and cohesin domains. And so these are protein-protein interaction domains that facilitate joining together of these enzymes. So for instance, this Ani9 here can have its docrin domain interact with the cohesin domain from Ani4, and then these can form a complex together. And then there's also non-enzymatic components that can add other functionalities, including other carbohydrate binding modules or these cell wall anchoring motifs for localizing the enzyme complexes to the surface of the bacteria. And then they also have some free enzymes as well. So we have these two different resistant starch degrading bacteria that we know of, and we can see some significant differences between them and the non-resistant starch degrading bacteria. But one of the interesting things is, okay, so we've got these two different bacteria, and then we've seen that we have a number of different types of resistant starches. Do these bacteria equally degrade these different types of resistant starches, or is one better at some, the other better at others? Or are they in constant competition for all of these? So one of the things that we've done is performed in vitro fermentation with resistant starches. So what we've done here is we had 10 people that we recruited, and we got fecal samples from them. And then we divided up those fecal samples, and then with the same fecal samples as starting material, we looked at whether they could equally ferment all these different resistant starches to butyrate, or does each person's microbiota respond in a different way? And do the different resistant starches select for different resistant starch degrading bacteria? And what kind of interactions are happening between those resistant starch degrading bacteria and other bacteria in the microbiota, particularly the butyrate-producing organisms? And so what we have here is, so on the y-axis, we have all the different starches that we looked at. So the media that these were grown in were all the same, except for we substituted in different carbohydrate sources. So that could be amylopectin or regular cornstarch as sort of our non-resistant starch controls. Or we have different sources of resistant starch, and these could be different types of resistant starch, or different botanical sources of the same type of resistant starch. So for instance, we have potato starch here, which is one of the classic sort of type 2 resistant starches with that B-type crystallinity. But we also have high amylose maize starch here. And so while maize starch is normally a non-resistant starch, if you have mutants where they make higher amounts of amylose, it changes the crystal structure to that B-type, and now it is a resistant starch. We also have a variation of this, the HAM4 here, where it's that high amylose maize starch that's been chemically cross-linked to make it even more resistant. And then we have some RS3 types here, some other RS4s, tiger nut and banana, which should also be type 2 resistant starches as well. And so what you can see here is that, and then on the x-axis, we have what was the different fecal samples that went into this. And what you can see is each fecal sample basically had its own pattern of butyrate production. So the blue means that there was an increase in the butyrate. The stars inside means that it was a significant increase. And if it's red, that means there's actually a decrease in butyrate relative to the control fermentation, which did not have the starch added to it. So even if we don't add any starch, there's enough nutrients in the base media that we at least get some butyrate production. And it turns out that with some of these, adding some of these starches in there shifted the fermentation away from butyrate to other substances like lactate, for instance. And so you can see that there's different patterns. Everyone that you looked at, though, even if they weren't very good overall at producing butyrate, always had at least one of these resistant starch sources that showed an increase in butyrate. If you look across the starches, the one that was nearly universal, almost, not quite, there was one that didn't reach statistical significance, but our banana flour was the best, or at least the most consistent in increasing butyrate production. Some individuals had butyrate production across most of the starches. Some had it almost with none of them. And they all had different patterns. The only ones that are kind of similar here are the TO4 and the TO5. And you can see there's still some slight differences, but generally speaking, pretty similar. Well, it turns out that those two are actually from the same person, just several months apart. And so what you can see that there's a consistency in this response in terms of butyrate production over time within a person, but it differs quite a bit between people. If you look at this TO9, you see that they produce tons of butyrate across all the conditions except for just the amylopectin. Even the cornstarch was a good source of that. So was there anything particularly different about the microbiome of this person? It turns out that most people have both Rhuminococcus bromii and Bifidobacterium allocentus in their microbiota. Some just have Bifidobacterium allocentus, some just have Rhuminococcus bromii. This was the only person in this study that only had Rhuminococcus bromii, and interestingly, they were the one that produced the most butyrate overall. Now, if we look at overall across all individuals, what the different starch treatments, which bacteria they selected for, again, each one had its own sort of set of bacteria that it enriched for. If we look at particularly Bifidobacterium adolescentis, we can see that it was actually enriched on most of the different starch sources. If we look at Rhuminococcus bromii, here we can see that it was a lot more limited what it was enriched on. The ones that the Bifidobacterium adolescentis were not enriched on were the high amylose maize starches. And these were some of the strongest ones for the Rhuminococcus bromii. So it does seem to some extent that there is this competition going on if both of them are present. But as we saw with this individual, you can get different results overall if only one of them is present. And then there's other bacteria that differed quite a bit. If you look at one group, the Roseburia are one of those important groups of butyrate producers. You can see that they were most enriched on those high amylose maize starches, although they were on some other ones too, like the retrograded starch as well. Now, if we look at it from the point of view of which bacterial species were correlated with butyrate levels overall, what you can see is here that the strongest associations were between different Bacterium prausnitzii strains and butyrate production. And that's not that surprising. They're one of the best known and most important butyrate producers in the gut. Interestingly, the relationship's not quite as, it is strongest if you only look, if you take the subset of treatments that had the most butyrate production, then you get strong associations within those treatments to butyrate levels with Bacterium prausnitzii. It's still significant if you look at the low butyrate producing conditions, a little less, but still pretty significant. If you look at, say, Bacterium rectali or Roseburia feces, they're really only significantly associated with butyrate levels in the high butyrate producing conditions. And so it suggests that maybe the Bacterium prausnitzii are kind of staying about the same across a wide range of conditions, and at certain other butyrate-producing bacterium, like Bacterium rectali and Roseburia, that are really governing the changes in butyrate levels that we see. Then if you look at these resistant starch degraders, there was interesting relationships. So neither of them were significantly associated with the butyrate levels overall. So bromii here, you can see it is a positive relationship, but not statistically significant. If you look at Bifidobacterium adolescentis, it just got up to being significant, but negative. So it seems that having a bromii-dominated fermentation is more conducive to butyrate production. If you look at which other bacteria Bifidobacterium adolescentis is associated with, it is associated with some butyrate producers, most notably like Anerostipes hydrus, which is one of those lactate to butyrate converting bacteria. So it's possible to get good butyrate production with a Bifidobacterium adolescentis dominated fermentation, but it relies on there being enough of these types of bacteria that are able to convert the lactate to butyrate. If you look at bromii, it has a lot more strong connections with a wider variety of butyrate-producing bacteria. So including those Faecalibacterium prausnitzii, but also strong with Eubacterium and Roseburia species as well. And so we think that when you have Ruminococcus bromii dominating the fermentation, it doesn't guarantee that you get an increase in butyrate, but it's more likely due to these strong connections to these other butyrate-producing bacteria. Now, coming back to that competition between the resistant starch degraders on these different starches, there was an interesting recent study by Noguera et al. Where they looked at competition between Ruminococcus bromii and Bifidobacterium adolescentis on potato starch granules. And when they do their fermentations, for instance, with different subjects, you can see that the people that had Bifidobacterium adolescentis, when it was present, over time, they came to dominate the cultures. You only saw increases in even E. rectali and Ruminococcus bromii when they were ones that were lacking the Bifidobacterium adolescentis. Otherwise, it seemed to suppress their growth. If you looked at the starch granules themselves, even if you started out the culture with a large amount of Ruminococcus bromii, what would happen over time is the Bifidobacterium adolescentis would displace it from the starch granules. So if you look at these potato starch associated columns here, looking at time 0, it's mostly Ruminococcus bromii because there was a lot more of that added. But then you go over time, after 6 hours, it's about a 50-50 mix. After 24 hours, it's almost all Bifidobacterium adolescentis colonized to those granules. So it's in some way able to displace the bromii. So it doesn't even just beat it through colonizing it initially, it's able to displace already established Ruminococcus bromii from those potato starch granules. Now, this is the results. That's the results of some in vitro work. We were interested to see if we could see the same sorts of things in human studies as well. So the first one was just looking at what if it was just a really low dose of potato starch that was coming into the diet by just replacing one refined grains dish per day with one potato containing dish per day. So this really only added about 1.7 grams of resistant starch per day. Would we be able to see changes in the gut microbiota? And so the answer was yes and no. So we didn't see that increase in butyrate happening when people switched to the potatoes, but we could see some changes to the gut microbiota. And so particularly 2 species, Roseburia feces and Hungatella xylinolytica, which were already fairly high at baseline, but increased even more on the potato treatment and then decreased a lot when they were switched to refined grain treatment. And note, in the baseline, the people were allowed to eat potatoes, but in the refined grain, they were not. And so you can almost think of this more as what happens when you take potatoes away from the diet. and you see you get a big decrease in this butyrate-producing bacteria. And actually, we had some participants the same between the human trial and that in vitro fermentation trial. And if you look in there and compare Roseburia feces levels between them, you can see that in both cases, there was a lot more in the potato versus the refined grain condition in the blue here. And then in vitro in red, between the control and potato starch, you still see an increase, but not as dramatic. But where you do see a dramatic increase, actually, is if you go to retrograded potato starch. So things like cooked and then cooled potatoes, like in a potato salad, or even leftover potatoes that have been sitting in the fridge. You get a reformation of that resistant starch, and that was even more selective for that Roseburia feces. So we could see something there, but we also wanted to look at a high dose regime. So whereas that one was only about a little less than 2 grams of resistant starch per day, this was with 20 grams of resistant starch per day. And this one, we've completed the trial, but we're still in the data analysis phase. And so I'm just showing you a subset of 10 individuals here and what happened to their butyrate levels. So the same as before, where the blue is an increase and the red is a decrease. And what we see is, compared to the control, just regular cornstarch, we have high amylose maize starch. The versified is that type 4 modified high amylose maize starch and the potato starch. And so we can see that some people get big increases on the potato starch, others do not. You can see, like, between these two individuals, this one just got that big increase on the potato, but not on the others. This one got a decrease on the potato, but good increases on both of the high amylose maize starches. And then some people had increases on all 3. And so we see this similar type of inter-individual variation in the patterns of response. There was, unfortunately, still some individuals like these two that didn't see any increase on the resistant starches, though interestingly, compared to baseline, they did see an increase on the control starch. So it's possible that even these slowly digested starches could have a positive influence on butyrate production. And then we were also interested in what happened to these resistant starch degrading bacteria between these. And similar to what we saw in vitro, on the high maize starch, we got a big increase in Rhinococcus bromii, but not Bifidobacterium adolescentis. And then the opposite on the potato starch, big increase in Bifidobacterium adolescentis, but not in the Rhinococcus bromii. Interestingly, with the versified, so this type 4 modified high amylose maize starch, it was not an increase in either of them. It seemed to be a different Rhinococcus, unidentified Rhinococcus species, actually a mixture of a few different ones. Also, to some extent, Rhinococcus bromii, but it's not dominated by bromii the way that just the regular high maize is. There seems to be a wider variety of these Rhinococcus, and perhaps these other species, unidentified species, are also resistant starch degrading organisms. And in both cases, we did see increases in Eubacterium rectali in those ones, but not in the potato starch condition. And so again, the design of this is a crossover trial. Both of these trials were crossover. So it's the same individual that is consuming each of the starches. So we can see within that very same microbiota what's happening when they encounter these different resistant starches. And so we can conclude that all resistant starches are not equal for all people. Yes, there is some people that respond pretty similarly to all 3 of the resistant starches, but most people have different responses in terms of how much butyrate is produced depending on what type of resistant starch they get. And so we think that points to, we need to think about having more targeted dietary approaches based on our gut microbiota. So the best resistant starch for one person might not be the best resistant starch for another. Someone might do really well with potato starch, but others might not. And then they need to switch to a different type of resistant starch. Others, it may not matter. So we're trying to think about ways of defining these, similar to the enterotype concept in the gut microbiota, where we can classify people based on their microbiota composition. Can we use that to say what type of resistant starch or what type of dietary fiber it would be best for you to be eating? Okay, and so with that, I will end my presentation. I just wanted to thank a few people. So the various people working in my lab, I particularly focused on some of the work of June Teichman and Peter DiMartino. And then much of this has been done in collaboration with people around the world, and funding sources here so far at Penn State have been the American Heart Association, US Department of Agriculture, and the Pennsylvania Department of Agriculture. And so thank you for your time, and I would be more than happy to answer any questions.
Oh, great. Thank you. Thank you very much, Darrell. If any attendant has a question, please, this is the moment. Can write the question in the chat, or also can open your microphone to do the question, please.
I have a question, Dr. Darrell.
Yes.
Thank you so much for your remarkable presentation, for sure enlightening. I'm a PhD student. I'm working with in vitro colony fermentation. And I was wondering, how do you determine 10 fecal donor for your study? Do you use a statistical tool for that?
How do we determine how many donors we need?
Yeah, how many donors you need? Yes.
Yeah, I'd say that is something that we are still trying to establish. Because, so when it comes down to is that you need to know how big of an effect size that you're expecting to see and how variable it is. So the effect size can be relatively strong that we've seen with this butyrate production when we add in big doses of resistant starch, but it's much less if we're trying to do the low dose of resistant starch. So we are still trying to kind of calibrate what those expected effect sizes are that we're going to see, which is going to partially determine how many replicates we need and how many different fecal donors we need. The other, on the variability, that's really an even bigger question, because like we saw, we have so much differences between people that what we were hoping was that with 10 people, we could maybe divide them into, 3 categories of what they're, and have within those 3 categories, the responses be the same. That was not the case. So it would seem that for a study like we did there, if we really wanted to get down into what were the commonalities and group people together, that we would need to go considerably larger than that. So we don't know how many. We've got another study ongoing with 20 people now, and we will see how that one goes. 20 people, and then it's not with as many different, in this case, it's only with 4 different substrates. So we're hoping that we will see more consistent patterns in that case, but we don't actually know how many different people we need to look at yet.
Thank you. Can I ask you other question?
Sure.
Do gut microbiota compositions change based on the ethnicity? I mean, for example, Bifidobacterium and Ruminococcus are present in all gut microbiota?
Yeah, that's a really good question. So I don't think this, for Ruminococcus bromii and adolescentis, I'm not aware that this has been looked at in enough detail to see. In the studies that have been done on prevalence, unfortunately, they've mostly been focused in the United States, for instance, with like the Human Gut Microbiota Project. There have been studies with, say, Bacterium rectali that have looked a little bit wider across the world, and they still see that you can find Bacterium rectali in every population that they look at, but it's different strains. So there can be geographical changes in what strains are present, and then there can potentially be differences in the capabilities within those strains across those geographic locations. Now, all of those Bacterium rectali across those different places seem to be butyrate producers, but they do have a little bit of differences in their starch-trading enzymes. They tend to have them, but there's some that don't. There's actually some strains that don't have those starch-trading enzymes. And so the conclusions that we reach might not be applicable necessarily to those ones. There's been a little bit of strain variation looked at in Ruminococcus bromii and Bifidobacterium adolescentis, but not enough to draw strong conclusions. They're probably all resistant starch traders, but maybe not all of them. And we also know that there's other closely related Bifidobacterium species to adolescentis that are also resistant starch degraders, but again, not all the members of those species necessarily. And as I said there, we highly suspect that there's other closely related to Ruminococcus bromii species and strains that are probably also resistant starch degraders. So we do have some work going on in my lab trying to isolate some more of these and look at them. But, you know, we are in State College, Pennsylvania, probably not representative of much of the world outside of Pennsylvania. So it would be nice to look at, once we get really good procedures established for isolating these bacteria, to start getting samples from a wider variety of areas and do something similar as to what's been done for E. rectali for these resistant starch degraders too, because I think that's a really important question.
Hello, Dr. Darrell. I have two questions. Thank you for your interesting presentation. My first question is, why did you choose banana flour? And the second, could you consider a different, non conventional source for resistant starch, like mango, for example?
We're interested in, so, okay, I'll answer the second question first. We're interested in all kinds of sources of resistant starch. It's more or less what we have available to us. We've looked at a little bit and are planning to look a little bit more. Starch from cocoa, for instance, seems to be somewhat resistant. We are looking at type C starches, so the ones from legumes, which have this mix of the A type and the B type. They don't seem to be as resistant overall as the true B-type starches, but they're more resistant than typical corn and wheat, for instance. But, you know, and then we have some collaborations with some companies where they give us their resistant starches that they've developed, but there's lots of people developing different ones and different potential sources. So we're interested in them, but there's quite a lot out there to look at. So maybe someday for some of them, but I don't know. In terms of why banana flour, it's one that's been reported. So it has to be green banana flour, and probably more likely from plantains. Because as the bananas ripen, they start converting their starch to a more digestible form. So it has to be green banana flour. And we were interested in one because it's something people use as a health food type thing. And we knew that it had another kind of type 2 resistant starch. So one of the things we're interested in is we have potato, we have high amylose maize starch. They're both type 2, but we can see that they have very different impacts on the gut microbiota. And so we wanted another type 2 to include in that. And interestingly, it was a bit more universal than the others and didn't strongly select for either Rhinococcus bromii or Bifidobacterium allocentis. They both increased, but not by huge amounts that we see on some of the other ones. And maybe that's part of the reason why it was more universal. It didn't exclude one or the other. It kind of allowed both of them to work. So yeah.
Thank you for your answer.
Darrell, we had some question in the chat.
Okay.
Please.
Would you advise to consume banana in a daily or weekly basis? So again, just want to emphasize that that is the green bananas. So if you want to eat an unripe banana, people don't tend to like those as much, but you will have more resistant starch in there. And I guess I wouldn't, at this point, advise anything necessarily. You can try it if you would like and see how it makes you feel. Some people can get gastrointestinal distress from eating green bananas, for instance. If it's causing you pain, definitely don't do that. But if you eat it and it's tolerated well, you could. But at this point, the data is too preliminary to recommend doing something like that. We're hoping that we will be able to get a full clinical trial where we investigate this in more detail, and then maybe we can start making some recommendations. Does the butyrate production across the starch source and the donors depend on the donor's usual diet and its effect on the gut microbiota? So probably yes. So when we're doing the human trials, we are not fully controlling. We record what people are eating, but we're not telling them this is the diet that you have to eat. That last human trial, that one went on for 6 months. That's a really long time to tell people exactly what they need to be eating. So they had a free diet other than the resistant starch that we were adding. Now, we are still in the process of trying to analyze the effect of other dietary components on that. I'm pretty sure there's going to be some, but when we add 20 grams of resistant starch, that's probably enough to overcome most of the other dietary changes. When we had only 2 grams of resistant starch, now their diet more dominated the situation. So it depends on the dose, probably. In the in vitro case, yes, they had different things that they were eating prior to donating the sample, but when we were doing the actual fermentation, it was all the same media plus the different resistant starches. So kind of like they were on the exact same diet, if you consider the media to be the diet in the in vitro case. And so in that way, the in vitro experiments can be complementary to the human or, say, animal model trials where there's other factors at play. You can really, in the in vitro case, really just drill down on the effects of the microbes themselves, because there isn't those host influences there. But in the end, we do need to try and take those into account in other types of experiments. I think that's all the questions in the chat.
Other question is Guadalupe Montserrat, Darrell. Did the banana flour have any previous digestion?
Oh, sorry. Sorry, I missed that one. So in that in vitro trial, all the resistant starches, they went through a simulated intestinal digestion. We did not do that with the amylopectin because there wouldn't have been any of it left in there for when we actually did the fermentation. So those ones, the non-resistant starches were not previously digested, but the resistant starches were, yes.
Dr. Darrell,
Yes.
I have a doubt about the starch-resistant enzymes.
Okay.
Are they attached on the extracellular surface of the bacteria, or where are they?
So mostly they're on the surface of the bacteria. Bifidobacterium adolescentis seems to secrete them to just be free-floating out there. Ruminococcus bromii, there's some that are not cell-associated, but most are. And then Ubacterium rectali and the Bacteroides that I showed, those are all cell-associated.
Thank you. And the last question, do butyrate production occur in the proximal column or on distant column?
Well, it occurs everywhere. So it depends on what is the fiber coming in as to where there's the most of it. So things like inulin, for instance, or fructooligosaccharides or other kind of soluble fibers are going to be more the end of the small intestine / cecum / start of the large intestine is where all that fermentation is going to be taking place. Other things, resistant starches are a bit more shifted towards the distal colon because they take longer to degrade. Now they start getting degraded earlier, so there is butyrate production still happening in the proximal colon. But other more insoluble fibers tend to be later. There's usually detectable across, but depending on the fiber, there can be a peak at different locations. So like I said, the peak for like inulin would be more the proximal colon, whereas the peak for most resistant starches would be the distal colon.
Thank you so much again, Dr. Darrell.
You're welcome.
Darrell, I had a question.
Yes.
What do you think about what is the role of food matrix in the starch digestion when I do fermentation in the colon?
Yeah, it's a good question. So we've only looked at this a little bit. So the, oops, hold on here. We'll go back to, when we did this with the retrograded potato starch, we did it in two ways. One where we either extracted the starch from the potatoes first and then made the retrograded potato starch, or where we did it in whole potatoes. And so if you look at the difference here as to now, of course, this is still in vitro, so you don't get the full effect in humans. There was maybe a bit more butyrate being produced from the whole versus the extracted, but pretty similar patterns overall. We do suspect that it's an important factor because, you know, the food matrix can sort of act as though it's making it a type 1 resistant starch, where you're blocking access to the starch by the enzymes. And so, you know, that could potentially shift it to later in the intestinal tract where it can be digested, compared to if you just took in a starch powder by itself. But, you know, honestly, that's not been investigated enough at this stage. So I think that's an area that needs to be investigated more, both for resistant starch and for other fibers as well.
Okay, thank you. Thank you very much.
You're welcome.
Any question? In the chat, Hector Adan.
Oh, let's see. Have you tried to evaluate type 5? We have not done any type 5s at this point. We do have someone in our department that kind of makes them, not from the perspective of making them resistant starches, but more from the perspective of making like host-guest inclusion complex type things for delivering different compounds. But we could look at them from the point of view of them being resistant starch. But we have not done it yet. What would we expect the results to be? It's a good question. If I were to guess, I would say that it's going to behave like it's somewhere in between a regular cornstarch and a high amylose cornstarch, so that it's more slowly digested than a regular cornstarch, but not quite as slowly digested as the high amylose. And I feel like that rate of digestion, I didn't really talk about this because we don't have really good data on this yet. But people have looked at this a little bit, that the rate of digestion might be one of the important factors in determining what is the profile of fermentation products that you get at the end. And that one of the things that might favor butyrate production is having a slower fermentation time course. So, but that there's probably all kinds of exceptions to that. But so that would be my guess, but we have not looked at it.
Okay, thank you. I think so. No more questions. Mario, can you give the certificate to Dr. Coburn, please?
Okay. Thank you, Doctor. We are going to present the certificate. One moment. Okay. Instituto Politécnico Nacional, Centro de Desarrollo de Productos Bióticos, award the present certificate to Dr. Darrell Cockburn, Department of Food Science, the Pennsylvania State University, for his participation as a speaker with the lecture entitled The Dietary Preference of Resistant Starch, Degrading Bacteria in the Human Gut, present virtually at the seminars of the doctoral program in development of biotic products, Yautepec, Morelos, 2 de mayo del 2022. Atentamente, la técnica al servicio de la patria. Doctora Gabriela Trejo Tapia, directora, doctora Perla Osorio Díaz. Muchísimas gracias, Dr. Darrell. Thank you very much for your excellent lecture.
Thank you very much. It was a lot of fun and lots of great questions. Really appreciate it. Thank you.
Thank you very much, Darrell. We are very happy to participate. I appreciate it.
Great.
Thank you.
Thank you.